Next, I've got massive respect for this sort of work because I'm sure it's something I could never have attempted, but mathematical modelling of the structural diversity of the cardiolipidome. Markus Keller's come from Innsbruck to tell us about that, and thank you very much.
Yeah, hello. I want to take the opportunity to thank the organizers and the Barth Syndrome Foundation to make it possible that I can attend this amazing conference here and to present some of the preliminary work that I have been able to establish in the last few months. First of all, the thing that interests me throughout already my whole career, in which I'm working on different lipid metabolizing pathways, is how the cells are able to establish in a quite specific way the structural diversity of different lipids that are present in the membranes, but also the structural diversity of special kinds of signaling lipids I was working earlier on. And one thing that I find very interesting about mitochondrial lipids is that you have this different kind of lipid pools, like for example the mature cardiolipin pool, which is well characterized by the different acyl chains that are connected to the polar head group of cardiolipins. And when you think about the other lipid pools that are also present in the same compartment, like other phospholipids, then you can see the different enzymes that are responsible for the de novo biosynthesis of cardiolipins and the enzymes that are responsible for the remodeling process as a kind of a link that are establishing an interaction between the phospholipid pool in mitochondria and the cardiolipin pool, as well as with the remaining cellular lipid pool. And the abundance of these enzymes and their specificity, and also the specificity of transport processes that are trafficking different kinds of lipids with certain specificity, is then responsible for the structural diversity in terms of the length of side chains and the number of double bonds of acyl side chains in these different lipid pools. And for studying these effects, what is affecting this gain of specificity, for example, in different tissues, of course, you need a methodology in which you can, as good as possible, assess the structural diversity within these different lipid pools. And this is with which I started beginning of the year to establish a method for that, and what I want to present today. So what I went for is a very classical liquid chromatography mass spectrometric method in which I construct or establish a gradient in which I elute the different cardiolipins in a time-dependent manner from the column. You see here the mass-to-charge ratio. We are quantifying singly charged cardiolipins, and in a retention time-dependent manner, they elute from the column in a really nice pattern where you have cardiolipins with the same number of carbon atoms, but with different numbers of double bonds eluting always in these nice lanes here. When you focus now on only one of these peaks that is eluting, like for example this peak here, the 1428, then you immediately see that this is not one single species that is defined by this mass, but you have this double peak, which is actually not a double peak, but there are different species inside this peak which have the same mass-to-charge ratio, so they are isobaric, but they have different distributions of different acyl side chains along the 4 possible positions of a cardiolipin. Sorry. And in order to get more structural information about all these individual cardiolipin species that are eluting throughout the gradient, one can perform fragmentation analysis, or MS2 spectra, by picking one of these peaks, bombarding it with nitrogen atoms in this case, and then measuring which kind of fragments are established during this process. And for cardiolipins, this is really well defined, which kind of fragmentations you get by bombarding. So I apologize for this shift here of the red lines, but along the backbone of the cardiolipin molecule, there are certain positions where we get fragmentation, and these positions are always the same no matter what kind of acyl side chains you have substituted here at these 4 different positions. What you get is a very specific fragment pattern. So this is one of the spectra that is recorded. And the different fragments that we measure originate from the different side chain compositions that we have on both arms of the cardiolipin molecule, and also from the different from the symmetrical or asymmetrical distribution of these side chains. For example, in this case, we have one fragment that is coming from cardiolipin that has 2 C18 chains on one side and 1 C16 and 1 C18 chain on the second side. Additionally to these main peaks here, we have minor satellite peaks that you maybe barely can see on this projection, but these originate from the fact that when you look on the distribution of double bonds between the 2 sides, you can have either, for this species, 2 on the left and 2 on the right side, or you can have 3 on one side and 1 on the other, and you will still get the same isobaric species. Of course, for analyzing one of these spectra by hand, it takes several minutes to attribute all these peaks, but it is possible to do that. But for the run that I showed you before, I have about 2,000 to 3,000 of these spectra, and it's a lot of work to annotate a single spectra of that. So that's why I wanted to go for a mathematical solution in which I can automate this process, not being forced to spend weeks and weeks on analyzing one single sample. And in order to automatically analyze this fragment spectra, you have to get this into a mathematical form, and I'm trying not to go too much into the detail. Basically, you have to formulate a function or a set of functions that is defining your problem, which I do here by this system of equations where I have on the left side basically the theoretical possible fragments, so all different fragments that could possibly be produced by fragmentation of all different kinds of cardiolipins. And on the right side, I have the actually measured fragment spectra profile. And what this mathematical modeling solution that was established here does, it is minimizing the distance between the theoretically possible fragmentation space and the actually measured spectra. And by minimizing this, we get at the end a profile of different cardiolipin side chains that are most likely to be present in this fragment spectra. And the distance that, so just some details, the distance measure that I use is a Euclidean distance, but also other distance measures work. I use a quasi-Newton minimization algorithm, but also other minimization algorithms work. However, this was the fastest. So I can normally, for 3,000 spectra, it takes 5 minutes to analyze. So how does the output look like? What do I get from one sample that is measured with this method? Here I show you one lipid extract from cultured things. Sorry. One lipid extract from a murine macrophage cell line that I was interested in a while ago. What we get is the normal abundance profile of different cardiolipin species, as we have seen it quite often in this conference. And for each of these species that we have here, we get a long list of different acyl side chain species. So what we typically see in a normal sample is that the composition of side chains is very symmetrical, as is also already reported. So we have the highest possible symmetry in the substitution of these cardiolipin species under normal conditions, especially for the length of the carbon atoms. For the number of double bonds, so how they are distributed, the variability is larger, so there is more asymmetry in this species, especially when there are more double bonds present. One first experiment that I did to test this method, if it's broadly usable for different research questions, was to record a kind of a cardiolipin map of the mouse, where we extracted 15 different tissues from wild-type mice and analyzed for their cardiolipin composition. And what you can see quite nicely here, only on basis of the MS1 data, is what is known that here the dot size represents how much of this one species is there. That heart is dominated by one species, whereas, for example, colon has very broad variability of cardiolipin species, which is more dominated by shorter side chains, whereas in the brain regions we have long side chains with a very broad double bond profile. And what you can do now with this detailed analysis data that I have shown you before is you can dissect the structural variability and the output that you get from the measurement into different components, as I did here for these different tissues. So you can either look on the total abundance, so how much cardiolipin is present in this tissue per protein, for example. I can also look on the broadness of the acyl side chain profile. So do I have one dominating species in the whole tissue, or do I have a lot of different side chain variability? Or how about the double bond broadness? Do I have a very narrow profile? Do I have a very broad profile? And by dissecting the variability in these different components, you can then nicely use these profiles and compare them to other data sets. Here, for example, I compared it to an mRNA data set from the same mice, which was published earlier, and looked if there is some gene regulation that is behaving, is co-correlating with these profiles that I have here. Basically, this analysis shows that the abundance of cardiolipins correlates well with energy metabolic pathways, which is not surprising because when I need a lot of mitochondria, I also have a lot of cardiolipins. The double bond broadness co-correlates well with brain function pathways. So there is nothing much surprising there, but it shows that how this data can be used. Yeah. So to conclude, I was able to establish a mass spectrometric method to quantify cardiolipins and to mathematically model the structural variability in these samples. And from this basic method now ongoing, we want to extend for monolysocardiolipins, also extend this for other phospholipids because it doesn't, it's readily extendable. Also for oxidized lipid species, we had a nice discussion yesterday about looking into that. And importantly, we can also do this for fragmentator of the fragments, as long as you know the fragmentation path. Yeah, and with that, I want to thank the people that have so far been involved in this study, and especially Gregor Oehmer, master students of mine, who did a lot of the work.
Thank you, Markus. It's a very elegant piece of work.
Yeah, as we discussed yesterday, I liked it very much. So did you try to play with the fragmentation energies and see whether the propensity of your fragmentation pathway changes in response to that?
I didn't show this here, but you not only have these three major fragmentation pathways that we have with our settings at the moment, but there are additional about 7 major fragments that you can get depending on how much energy you put into the fragmentation system. We optimize the energy for fragmentation in a way that we get this in a very reproducible way, because this gives us the most structural information. But by altering the settings, you could also generate the monolyso fragments and quantify those.
people have been trying to make the fragmentation stereo-specific. This is something that has never really been brought to a really applicable stage.
Yeah.
But I think your method could be useful in validating different fragmentation techniques that are available in better instruments than the one you have, of course.
Yeah,
you would
exactly.
eventually have to move up a little.
One problem with the monolyser form, it would be nice to use this information as well, but the sensitivity, we have simply sensitivity issues to get enough of the signal.
Very elegant work. I have two questions. The first one, do you have to use high resolution mass spec, or how many significant figures you have to have for your model?
Yeah.
And the second question, is it any way to extract information regarding double bond location on acyl chains?
Okay, regarding the first question, one reason for making this modeling approach was that I have a quite low resolution instrument which I can use. I'm happy about being able to have a mass spec available. Of course, this would be much better if I had a high resolution instrument for that. But this will only increase the accuracy of the fit and maybe generate a little bit more information, maybe making this MS3 spectra possible. But the principle of this modeling can be applied also for high resolution data. Regarding sorry, what was the second?
I was wondering if there is a way to extract information on double bond location.
Yeah, exactly. I think there was quite recently, unfortunately I forgot the authors, but there was a quite nice paper about double bond location where you photochemically cleave the double bonds at certain positions, and then by analyzing the fragments, you can determine where the double bonds were. You could also use this technique in combination with this, but I have not tried that. This was quite impressive work, and I can look up the paper for you if you want.
So I have a quick question. Did you look at the cardiolipin profile in macrophage? Because cardiolipin really play a very different role in the immune system versus in peripheral tissue.
So if the profiles are very different in macrophage, so what I also measured was fibroblasts and macrophages so far, and the profile in macrophages and fibroblasts are both quite broad. I was actually quite interested in seeing how different cell lines from the tissues where you have these very specific patterns behave in culture, because I have the suspicion that in culture you're also changing your profiles simply by the nutrients that you provide. So I'm not so sure. I'm at the very beginning of trying to understand what influences the broadness of this cardiolipin pulse, but I think the way how we are cultivating cells has a massive influence on that. And since this macrophage cell line is grown under specific conditions with a lot of glucose, with a lot of glutamate in this case, I think this can have an influence on how the lipidome in mitochondria in general looks like.
Yeah. So that was a really very interesting poster yesterday, and so congratulations with your results. So now for many people, I want to make it more clear that in tissues with highly diversified cardiolipins, just in the primary total spectrum, you can see hundreds of molecular species.
Yeah.
But they will have all the same mass. And of course, we don't know all the permutations and total amount of topoisomers.
Yeah.
So now, as you did these calculations, what is it on average, or so what is the number of topoisomers per each signal in the mass spec that you detected?
This varies quite a lot. So for
Yes.
some, there are only two major or one major species. For basically, the more double bonds you have and the longer the side chains are, the higher the number of isomers are. I would say you get. It's also really hard to get a clear cutoff. Is something that is there with 1%.
But on average, just to give a ballpark of numbers.
I would say for the high molecular weight brain cardiolipins, you have 20 or 30 different species, with the sensitivity that with which we can measure them. I would say about this number for the lower molecular weight cardiolipins, or for E. coli cardiolipins, it's much narrower, this number, for 1 or 2 or 3.
And then the second technical, I didn't understand your chromatogram. That was reverse or normal phase?
It's reversed phase
So
chromatogram.
do you see them top isomers separately for some of them?
No, the chromatography is actually designed to broaden the peaks that are eluting in a way that I have enough time to accumulate all this MS2 spectra. So I'm trying not to get very sharp peaks, but I'm trying to get broad peaks in order to gain measurement time, actually. Yeah.
Okay, Markus, thank you for explaining so clearly and honestly about your work. Well done.